Hi there. This is part 5 of the genomics series. This is part B of the bacterial genomes. So I'll finish up talking about some of the bacterial genomes, including cyanobacterium, a photosynthetic bacterium, and then I'll go on to Archaea. I won't completely leave bacteria out because once we talk about parasites and endosymbionts, I'll be talking about some other bacteria also with reduced genomes. The next genome I want to discuss is of a cyanobacterium, and it's Synechococcus WH8102. It's a photosynthetic bacterium that's widespread around the world. Here's a general picture of a cyanobacterium. It's a diaderm, that is, it has an outer membrane, a peptidoglycan layer, and an inner cell membrane, as well as a thylakoid membrane on the inside, and what are called phycobilisomes, which are part of the antenna complex for concentrating the energy of photons. And then inside of that, there are lipid droplets, as well as the DNA and ribosomes, etc., inside of the cell. Here are some diagrams of the antenna complexes for cyanobacteria and red algae on the left, and for plants and green algae on the right. So on the left, you have a phycobilisome in the middle of the photosystem, in this case photosystem 2. And there are phycocyanins and phycoerythrins, which react with the photons and then excite electrons and move those electrons down towards the reaction center in photosystem 2. In plants and green algae, it's a little different in that they have light harvesting complexes in both photosystem 1 and photosystem 2 for the same purpose, that is, for exciting electrons after being hit by energetic photons. Cyanobacteria, red algae, green algae, and plants all have two photosystems, photosystem 1 and photosystem 2. Down below here you can see some of the genes necessary for the allophycocyanin, phycoerythrin, etc., as well as the light harvesting complex genes. And that's in addition to all of the proteins needed for photosystem 1 and photosystem 2, which requires an entirely different set of genes than things like E. coli and other non-photosynthetic bacteria do not have at all. In addition to the antenna complexes and the genes and proteins needed for that, dozens of genes are needed for the light reactions, and about 24 genes are needed for the dark reactions. So another set of genes are needed that, again, are not present in things like E. coli and other non-photosynthetic bacteria. So in this case, you have photosystem 2 being hit by light, an electron that's been stripped away from water to form oxygen and hydrogen ions. The electron then moves down the chain through some plastoquinones, and then through cytochrome, plastocyanin, and then through photosystem 1. Light further energizes that, and so you have NADPH produced at the end of that reaction. And the protons go through an ATP synthase to make ATP from ADP. Then the NADPH and the ATP move over into the dark reactions, where they're needed to fix carbon dioxide into organic molecules to make sugars, starches, and other organic molecules. So again, dozens of genes to form each one of those proteins in each one of those sections are needed for this process. Here is the same process, just shown in a slightly different way. So you have the phycobilisome that's gathering the energy from the photons, and it's gathering that in the form of energetic electrons, feeding that down into photosystem 2, where you're getting splitting of water into oxygen and protons, and the electron gets shifted into the electron transport chain in the thylakoid membrane. The energized electrons are then shifted through a quinone, and then through cytochrome, through plastocyanin, and then through photosystem 1, where there's further energization of the photosystem, the electron in that photosystem. All of this acts to pump protons across the membrane, and then those protons flow back through the ATP synthase to make ATP. And then in the stroma of the chloroplast, that ATP and NADPH is used to fix carbon into organic molecules using the Calvin cycle. To add a little more complexity to this system, depending on the environmental conditions, the pigments will change in the cyanobacterium. So you can see on the lower left, that's under different environmental conditions, you'll get different pigments formed, all of them to optimize not only photosynthesis, but also some to protect the organism from too much damage due to the irradiation from the sun. So here's a description of what is in the genome of Synechococcus WH8102. It has a genome size of 2.4 million base pairs, about 2,800 genes, including 2 ribosomal RNA gene operons and 44 tRNA genes. Now you'll notice that having 2 ribosomal RNA genes would not allow it to divide every 20 minutes, but often these become polyploid, so they at times can reproduce even faster. This is one of the photoautotrophs, which fixes carbon using photosynthesis and the Calvin cycle. Most cyanobacteria have oxygenic photosynthesis, that is, they split water into oxygen gas, electrons, and protons. There are a few that are missing one of the photosystems and are anoxygenic, but this one's oxygenic. The protons are then shuttled through protein complexes in the inner membrane. Then the electron energy is used to move protons, and the protons are ultimately shuttled through ATP synthase to produce ATP, and the electrons and protons are added to NADP to produce NADPH, and both of these are used in the Calvin cycle to fix CO2 into organic compounds. So already you can see that would take a large number of genes in order to accomplish all of that. So again, this is a gram-negative picoplankton. That is, these are actually small cells, and 25% of the organic production in the oceans comes from this group of cyanobacteria. It can use nitrate, ammonia, or urea as a sole nitrogen source. It has phycobiliproteins, phycocyanin, allophycocyanin, allophycocyanin B, and phycoerythrin. These are the pigments that then focus all of that photon energy into electron energy to feed into the photosystem. It's marine and occurs in all oceans, although it's limited in polar regions. It doesn't like really cold water. Genetically variable, since there are some that have genome sizes of 2.4 million bases, 2.9 million bases, and over 3 million base pairs. So it's a sort of like E. coli. It's probably multiple species. It has a circular genome, and so this is the one that's 3 megabases, Synechococcus PCC7002, slightly different genome. And then the outer colored rings indicate genes of different functions on one strand versus the other strand, so there's 2 rings there. And a lot of them are related to photosynthesis, but not quite as many as you might think are photosynthesis genes. So it has a lot of functions. It can live in a wide variety of ocean environments. So here's a pie chart of the breakdown of the different categories of genes. Only 4.2% is devoted to photosynthesis. So if you take that out, some of the proportions look somewhat like an E. coli. So there's 13% devoted to energy metabolism, 10% to the cell envelope. There's 8% to DNA replication and nucleic acid synthesis, about 6% to transcription, and about 11% to translation. And as with E. coli, there's still quite a bit, about 10% to transport and binding. That would be intracellular transport and binding and communication, as well as transport and binding to things going from the inside of the cell to the outside of the cell and vice versa. The next bacterium I want to talk about is Pyralula. It is within the planktomycetes, and if you remember, this is the group that has internal membranes, including a membrane in Gemeta, Bacerial globus, which looks like a nuclear membrane. So this is an interesting group. They also have fairly large genomes within this group. So on the right is Pyralula, and you can see the internal membranes within in those cells. It also has fairly prominent flagella. So that's one with internal membranes that differ from Gemeta Bacerial globus. That's the one that has these membranes that look like they form a nucleus with a double membrane structure and nuclear pores, and the DNA is on the inside with ribosomes inside and out. So that one's the one that looks like a nucleus, but this group has several different subgroups within it that have different membrane systems, but all have internal membrane systems. As I indicated, these have larger genomes, so that Pyralula has a genome size of about 7.1 million base pairs, and Gemeta Bacerial globus has a genome size of about 9 million base pairs. Now, these are approaching genome sizes that are close to those found in fungi. So these are, pretty good sized genomes with a lot of genes, 7,700 genes in this case for Pyralula. And a lot of those genes are involved in membrane structure and transport in and out of membranes, which you might expect for an organism that has internal membranes. So these are freshwater spherical bacteria. They can be aerobic and anaerobic, so they can adjust to quite a few different environmental conditions, which takes a lot of genes to switch from one type to the other. Most genes are there for replication, transcription, metabolism, and those are closest to other bacteria. However, some genes are more similar to eukaryotic sequences, which is interesting because one of the theories is that this was this was the group that led to the formation of the nucleus, or the acquisition of a nucleus and internal membranes in eukaryotes. Multi-copy genes make up more than 25% of the genome, which is also more eukaryotic-like than bacterial-like. These are chemoheterotrophs. They have no peptidoglycan cell walls, which is unusual for a bacterium. The ribosomes are attached to a nuclear membrane, which is also strange for a bacterium, right? And the nuclear membrane is continuous with the cell membrane, and this is similar to a eukaryotic system. They have polar yeast-like budding cell division, so they look like sort of like a yeast as it buds out and divides. Has a large number of sulfatases, of which 75% are bacterial-like and 25% are eukaryotic-like. It uses these to scavenge carbon from sulfonated carbon compounds. Also, quite a few transposons, 81 transposons, again, a large number for bacteria. And the FTS genes are similar to tubulin genes in eukaryotes. And as I indicated, it has a high number of membrane targeting sequences, that is, signal peptides and things like that, possibly for maintaining internal membranes, including the nucleus-like structure in some of these species. So we already know that Archaea are closely related to eukaryotes, and probably an Archaea joined with a bacterium to form the first eukaryotic cells. And this planktomycete group is a good candidate for that initial joining to form a eukaryote, because it has internal membranes, transposons, a lot of eukaryote-like characteristics and genes. This is a diagram of the genome, and then the circles represent different gene functions and identification of different elements. The outer two circles are genes on one of the strands of the chromosome versus the other strand of the chromosome, and the functions are shown here. I've highlighted a few of those, including protein synthesis and destination, that is, a destination in the cell. So those are in higher proportion than in most bacteria. So you have the proteins winding up in very different parts of the cell. And the second one is transport, and that's transport across membranes, and in this case, multiple membranes. The next one down, circle 3, are for highly expressed genes. So those ones are expressed in high volume. The next one shows in blue things that are more closely related to eukaryotes. And in green, those genes that are more closely related to Archaea. So that's interesting. And then the fifth circle, those yellow bands indicate sulfatases, and that's for scavenging carbon from sulfonated compounds. And the final ring in there, circle 6, indicates regions of the chromosome that indicate lateral or horizontal gene transfers, clusters of genes that have been the product of horizontal gene transfer from other species. So next I'll talk about a few archaeal genomes, and these are more closely related to us, actually, to the eukaryotes, so that a lot of the systems in Archaea are systems that are similar to those in eukaryotes. So you can see by this graph that they have a narrower range of genome sizes than do bacteria. And it's actually a smaller group than the bacteria. Bacteria are a larger group, but on the same range, essentially, of genome sizes to bacteria. The first archaeal species that was sequenced completely was Methanococcus yanashii, and if you look it up, you'll find it under Methanococcus yanashii, but it was since then it's been renamed. These were isolated from thermal vents in the bottom of the ocean, so they're at high temperature. They grow at high temperatures and high pressures. And as you can see down here, they originate close to black smokers, which have a lot of sulfur compounds in them. And over here you can see they have a lot of flagella, so they move around within the water column. One of the reasons this was the first one to be sequenced is it has a rather small genome of 1.7 million base pairs, about 1,800 genes, 2 ribosomal RNA gene operons, 37 tRNAs. But it also has a couple of extra chromosomal elements, one that's 58 kilobases in length, which is a little larger than the lambda genome, with 46 genes, and then one that's 16.5 kilobases, which has 12 genes. 16.5 is about the same size as our mitochondrial genome. The core set of genes is similar to that of Haemophilus influenzae, a free-living bacterium, but they're all thermostable versions. That is, they have more cysteine residues in them and disulfide bridges. Many of the genes are more similar to those from eukaryotes than to bacteria, and I'll show you more about that in a little bit. Their temperature range is from 48 to 94, so you can imagine that they have some genes and some proteins that are stable under varying temperatures. Their optimal temperature is 85 degrees. They're also barotrophs because they live at high pressure on the ocean floor near thermal vents. They're methanogens, chemoautotrophs, and they reduce CO2 with H2 to produce the methane. And it contains many of the genes for fixing nitrogen, but not all, and that's still questionable. So they may have to gather their nitrogen compounds from other organisms near those thermal vents. They also contain histones. So bacteria have histone-like proteins, but Archaea have true histones. So there are histone genes. And the genes are somewhat midway between those of bacteria and those of eukarya. Here's the genome as it was reported in the 1990s, Methanococcus genacei. And it shows the genes on one strand versus the other strand. So again, quite a packed genome. So bacteria and Archaea have genomes where the genes are fairly close together. The little H-like things interior to that, I thought I'd show this the way it was first reported. Those actually represent all of the clones that were produced and then sequenced. When this was done, the automated sequencing was just coming into fruition. So most of this took like 5 years, and I don't know how many labs to do. Each one of those separate little H's was a section of the chromosome that was sequenced by probably one graduate student, and then they were all put together into the complete genome eventually. The lines interior to those represent the locations of the tRNAs and the ribosomal RNAs and some other repetitive elements as well. So here's a pie chart breaking down the functional groupings of the genes. 10% to energy metabolism, about 8% to methanogenesis, and then 11% to DNA replication, then 6% to transcription and transcription regulation, about 20% to amino acid synthesis and translation, so quite a bit, and 3% to cell envelope, etc. So at the time this paper was published, there was a lot of interest to trace down the origin of eukaryotes, and so Archaea were a big part of that theory of the origin of eukaryotes coming from Archaea. And this shows that to some extent. So there's a comparison between bacteria, and in this case it was E. coli, Archaea, Methanococcus, and eukarya. This was compared to Saccharomyces in this case. So everything that's bacterial-like is in red, everything that's eukaryotic-like is in blue, and purple is specific to Archaea. So for lifestyle types of genes, that is being unicellular, having transport across the membrane with the environment rather than another cell, etc., origins of replication, size of the genome, then Archaea looks similar to bacteria. But then if you look at the microtubules, then the mode of replication, transcription, translation, those sorts of things, Archaea and eukaryotes have much more in common. So from this you can infer or conclude that a lot of the replication, transcription, translation, and some other functions that you find in eukaryotes came directly from Archaea. And this likely originated in that first eukaryotic cell, which was the fusing essentially between an archaeal cell and a bacterial cell to become that first eukaryote. More recently, hundreds of genomes from various organisms, both those that have been cultured and those that are uncultured and known only by their sequences, were used to produce a large phylogram. And in this case, you can see the bacteria over on the left, and then there's a whole other group of organisms on the right, a candidate radiation, which consists only of groups of organisms that have never been cultured and weren't known about until these environmental sequencing methods were used. But to go down to the lower part, you can see that there is no real separation between eukaryotes and the archaea, again bolstering the theory that eukaryotes are just a branch of the archaea, that they came off of that branch. So many of our genes, or most of our genes, were inherited directly from an archaea or an archaeal ancestor. So this is just one of the very interesting things that you can discover when you examine these genomes, and especially large sets of genomes. One other archaeum that I wanted to talk about is this one, Sulfolobus sulfatarchus. A lot of archaea are extremophiles. This is one that lives in hot acidic pools or hot acidic mud. This particular one is was isolated from Italy. And one of the main reasons I wanted to talk about it is that, it lives in these hot sulfur pools and hot sulfur mud. This one was isolated from a volcano, or a hot pool from a volcano in Italy around Naples. And Naples in that area, which includes Mount Vesuvius on one side also, is a huge supervolcano, a huge caldera, which, if it ever explodes, it'll just obliterate that whole area. It'll be much more violent than Vesuvius. Vesuvius is just a tiny little subset of the entire caldera. One of the things also that was discovered with this archaea were archaeal viruses, and you can see those over there. So prior to investigating some of these archaea, it was thought for a while that archaea didn't have phages or viruses that attack them, but now it's known they do have them, although they look very different from bacteriophage. So here's a description of this genome. It's about 3 million base pairs, almost 3,000 genes, 4 ribosomal RNA gene operons, and 44 tRNAs. It's aerobic and grows in these hot acidic pools and mud pots where the pH is around 2 to 3 and the temperature is between 75 and 80 degrees centigrade. So this one's an acidophile and a thermophile, isolated from this volcano named Solfatara near Naples, Italy. Many of the genes are more similar to analogous genes from eukaryotes than bacteria, which again was expected because archaea are closer to eukaryotes than eukaryotes are to bacteria. Also, many bacterial chromosomes have only one origin of replication. This one has more than one, so this is on the way towards what eukaryotes have, which is multiple origins of replication. The replication mechanism is much more eukaryotic-like than bacterial-like. The proteins are thermostable, but they're not resistant to acids, which surprised people when they first started looking at this. But what happens is the cells maintain a large pH gradient, so they push the protons out of the cells and then import the protons through the ATP synthase, so they actually have an easy way to make ATP due to the large proton gradient. They also found viruses in the genome and found that archaea are infected by viruses. They found glycolysis, TCA, pentose phosphate pathway, all of the central metabolic pathways were there. It can oxidize sulfur. There's a lot of sulfur around there. And it can then make simple carbon compounds using that energy. And the proteins in the respiratory chain, on the other hand, are more like those in bacteria. Here is a pie chart of the different gene categories. 6.6% is devoted to energy metabolism. This is lower than a lot of other bacteria or archaea. And so it may be that since it has a ready supply of protons and ATP, and it has a lot of thermal energy, that it devotes less of its genome to energy metabolism. About 8% for DNA replication, and then for transcription, about 24%, which is very, very large. So it's probably a lot of transcription regulation, possibly due to the high temperatures. And then about 10% to amino acid synthesis and translation, and about 5% to the cell envelope. So again, there are some central biochemical pathways and functions that have common genes, and you find those in bacteria, in archaea, and eukaryotes. But then each one has some special functions, as well as some functional gene sets that are in higher amounts in different species. And that's what you find in the ones that are photosynthetic have an increase in those that grow in hot places, in high pressures, etc. You have some specialization in gene sets due to those different environments. And of course, the ones that can grow in many different environments also have larger gene sets because they have to switch from one set of environmental conditions to another. So that's the end of this section on bacteria and archaea, although I won't be abandoning our bacteria altogether, because when we talk about parasites, symbionts, and endosymbionts, I'll be talking about the bacteria that fit into those categories as well. But for now, next time I'll talk about parasites and endosymbionts. Talk to you later. Thanks.